with you. So let me just give a short introduction. So Lou Pambianco, well, just generally, the goal of this program, this was Todd Whipke's idea, maybe as much as a year ago, is to give everybody some information on what's happening with the university's efforts to allow faculty to take the research that they've done in their academic career, and if they think this can be converted into some sort of useful business enterprise to do the research that they need to do the business part of it in a separate facility and go on to start some sort of useful enterprise. So UCSC has gotten into this in a fairly big way in just recent years, and we're going to have a few speakers here tell us some examples of how this is done. So the key component here is this group called Startup Sandbox, and what that is, you will be informed of by the CEO of Startup Sandbox. This is Lou Pambianco. Lou got his education at Santa Clara University. He was a consultant for more than 20 years for various technical companies, consulting in their early stages development. He's also a very avid sailor, and so Santa Cruz is a good place for him. So Lou, why don't you take it away?
Well, thank you very much. And it's such a privilege and an honor to be able to present to you today, and I will get right to it by sharing my screen. Do I need permission, or can I just go right ahead?
Go ahead.
Okay.
Also, Lou, I'm going to mute everybody, and then so please unmute yourself.
Okay.
Okay, you're still muted.
Okay.
Good, we can hear you.
You hear me now?
Yes.
Wonderful. Let me just get things set up here. Okay. So I am the co-founder and CEO of Startup Sandbox, and I want to give you an overview of the Sandbox so that you can have a sense of what it is that we do and how we can support your efforts. Okay, let's see. I'm not getting, hang on just a minute. I'm attempting to move through, and it's not working, so let me try once more.
Good.
There we go. Now you can see me. Well, I don't know why it's not advancing my slides. Okay.
The message would suggest you're at the last slide. In fact, if I look at the left of the screen now.
Oh, well, you know what? That would tell me why I'm not advancing. Okay.
Thanks, Charlie.
There we go. There we go. All right. So we're a bioscience incubator, and here's a picture of our building at 250 Natural Bridges. And we're just down from the campus. And this is where folks come to validate their research, and we move them into the commercial product area as rapidly as we can. So it is UCSC's 501c3 incubator. We have both BSL-1 and BSL-2 labs. By the way, the initial funding for this came from President Janet Napolitano's AB 2664. And we completed it under budget and ahead of schedule. we immediately recruited and mentored and grew our first cohort of 7 wet lab and 10 dry lab companies. So wet lab, of course, are those companies that would need a lab space to do things in fume hoods and in incubators, et cetera. The dry lab companies could be a 3D printing or a bioinformatics company that don't necessarily need the BSL-1 and the BSL-2s. So we've had over 25 member companies since 2018. So it's quite a group, and I'll go through those later. We also created what we call the Founder Circle. So Judy Owen, who was our initial venture partner in the startup Sandbox, and I went out to our network of individuals, both in high tech and in the biosciences. And we have now over 30 Founder Circle members who engage with and help with many of our companies. They've contributed $250,000 to the Sandbox, which is a tax deduction for them. We are now launching the Sandbox Venture Fund, and that's headed up by Laura Oliphant, 15 years at Intel Capital. The companies that she invested in for Intel delivered over $1 billion to Intel during that period of time when she was active. So she is now the general partner, and we're launching an initial phase of the fund, which is a $5 to $10 million phase, followed by a $25 million fund. The purpose of this fund is to give each Sandbox member company, upon joining the Sandbox, a seed fund of $50,000 to $100,000, and with the rights to co-invest up through and including the A round, should that company continue to succeed. So we're really focused on developing, launching, and commercializing. And in talking with Chancellor LaRive, she said, we aren't a teaching hospital as UCSF is. So I'm really happy to see that you're focusing on these core areas of excellence that the university is known for. And certainly physical sciences, both biology and chemistry and biochemistry are included in this, as well as so many of the other areas of the university. But we're really pulling companies into the Sandbox, validating their ideas, defining the product, and formulating their strategy for success. And then we're launching those companies. And you'll see how quickly we're operating. And what we'd like to see is that these companies stay in Santa Cruz. And again, you'll see shortly that many of them have. So the lab is very familiar to many of you. PCR, the fume hoods, the minus 80, 20 freezers, the BSL-2 lab with its incubator, centrifuges, et cetera. So all of the things that a postdoc or senior scientist would need in order to be successful. So we have a formula for driving IP commercialization at the Sandbox. And this was created over a year ago with David Deemer, who's on this call, Todd Lowe, David Hausler, Harry Noller. And what we looked at was the key stakeholders at the university, from the Genomics Institute for the Center of Molecular Biology of RNA, the Institute of Stem Cell, nanopore sequencing, and more. There may be, coming out of these areas, a particular research that potentially could be commercialized. And we'll have a Sandbox program manager that will drive that IP through commercialization delivery to the open markets. And we're looking at creating capabilities such as sample management partnerships, bioinformatics core hosted at the Sandbox through David Hausler, CRISPR core, sequencing, robotic liquid handling, and 3D biomaterial printing. Some of these are in place, others will soon be in place. So we're really interested in elevating the UCSC research to the next level of commercial products and therapeutics. And when I say research, certainly those of you on the call today come from various areas of the university, and the IP portfolio in the Office of Research, directed by Scott Brandt and managed by Jeff Jackson, is very rich. And by using our commercialization team to partner with and leverage the unique leadership and IP of the university, we're bringing products to market very quickly. So let me give you just a little bit of background. I formed a company in the early 1980s called Corporate Development Partners, CDP. And we've helped early stage companies when in fact they were early stage. So an example would be Intel when they were a DRAM company and transitioning to microprocessors. They were on the stock market at the time at $4.25 a share, and they were being shorted. No one thought they would survive. The reality is they grew from where they were at about $1.5 million in revenue, $1.5 billion in revenue in those days, to over $35 billion in revenue just a few short years later. We built the software integration model at the architectural level of the chip. During those days, Intel was a hardware bigoted company. They didn't allow software to participate in the architectural design. So, of course, they've grown since, and we continue to work with them over the years in many capacities. Cisco, Apple, Sun, Compaq, all of these have been clients in their early days. At Apple, we partnered with Dan Eilers, who was the executive VP of corporate strategy, and went on to their software group at that time, which was Claris, and we built the Claris software strategy. FileMaker became the name of that for many years, and just last year, they renamed it Claris. We also worked with Sun in the early days and Compaq. Connor Peripherals was a core client, and we built a growth model for Connor Peripherals, which became known as the exceptional growth model. What does it take to grow from 0 to $1 billion in revenue in 10 years or less? They became the fastest growing company in American business history, and they were Fortune's Company of the Year. We're now utilizing those exceptional growth lessons with each of the member companies that come to the Sandbox. And that's in part how we've been so successful getting those companies into the commercial markets rapidly and giving them the horsepower to continue to develop and grow rapidly. So we're applying those processes, and we believe that these companies, which are now defining the first half of the 21st century, will be companies that will grow to unfettered growth and extraordinary profits. So Sandbox commercial launches. We've graduated our initial 6 companies in under 18 months. Those companies now are occupying over 20,000 square feet of commercial space in Santa Cruz, much of it on Delaware, 4,500 square feet in Silicon Valley. They've raised in excess now of $20 million, much of that Series A financing. They've hired 12 graduate and postgraduate UCSC scientists, more on the way. And now they're referring new member companies to the Sandbox. Sandbox successes in less than 18 months, dimensional bioceramics. They've received an offer for acquisition of their patented bone compatible division. UnNatural products. UNP has created a drug platform and is licensing that platform to pharma companies. Prime Genomics. Prime Genomics closed a $7.5 million Series A round, positioning themselves for a probably $30 million B round. And they're expanding their breast cancer diagnostics. Claret Bio, selling their DNA kits to pharma. Cruise Foam, Marco Rolande's technology. They've signed a distribution agreement for biodegradable foam packaging. Can you imagine getting rid of styrofoam? Well, they have the opportunity to do that. Aeroasis came to us from Florida. This is a wonderful young entrepreneur who created an elegant hydroponic capability for growing green leafy vegetables. You can put that in your living room and it's a gorgeous environment. Read it out on your smartphone, control it. They have a distribution agreement in Dubai for hydroponic growing appliances. These are some of the cancer therapeutic companies. Prime Genomics, UnNatural Products, UpRNA, and Claret Bio. And these are some of the Sandbox companies in the age of COVID-19. Incisive RNA is Bill Scott's company and Sarah O'Rourke. Incisive RNA has done remarkable work in directly targeting and destroying SARS-CoV-2 by killing the message. Pinpoint Science, coming from the work that Nader Pourman has done, CEO Lisa Diamond. Nanosensors on a medical device that's handheld that will give you a 30-second positive detection, 98% accuracy of coronavirus. Sanguine DX, COVID-19 antigen and serology test device. So all three of these companies currently are being assessed by the Gates COVID-19 initiative. I was able to put this package together and send it up to the CEO of the Bill and Melinda Gates Foundation, and he put it in the proper places, and they're currently being assessed and in their second round of due diligence currently. So 25 companies have participated at the Sandbox since July 2018. That's when our lab officially opened. I'll have to tell you a wonderful story, and I'll make it quick. When we first took the lease on the building at 250 Natural Bridges, we had 7 companies that wanted to be in our lab. And we said, my God, we're not going to have a lab until at least July of 2018. And then being startup individuals, we said, look, we have a break room upstairs, 200 square feet, tile floor, water source. Let's turn it into a temporary lab. So we did. The first individual in that lab was David Deemer. And he was testing his theory on whether, in fact, he could synthesize siRNA. Thank you, David. Following him was Prime Genomics and a number of other companies. But we have everything here from Zanskar, which is doing geothermal remote mining. They're currently looking at opening up a geothermal capability to extract rare minerals in a remote part of Alaska. So I won't go through all of these companies, but suffice it to say, there's some amazing technologies that have been at the Sandbox and are coming through the Sandbox. Our next cohort of Sandbox member companies, LinkRNA. So Dr. Kim, early cancer detection. Pulmodine, This is a company that will be coming to us from the East Coast, out of the University of Michigan. They're doing increased crop volume for both plant and fruit. Frontier Bio 3D tissue printing. They're also coming to us from the East Coast. Eugenics, amazing capability in anti-aging. Sensorium Bio, cancer diagnostics, and NanoHue, utilizing nanopore tracing and testing. We have a very solid pipeline of additional bioscience companies. And with the raise of the current Sandbox Venture Fund, we'll be able to put money into each of these companies that gets them started in the sandbox. By the way, for all of the professors on the call, what Laura Oliphant is doing is creating a special limited partnership that would be offered to the professors of the university for a small infusion on each of the professors' parts. The aggregation of those funds would become its own LP, which I think is a marvelous thing. So that's Startup Sandbox in a few short slides, and I hope that that has been helpful to you. And I will stop my screen share. And in the event that there are any questions, Barry, do you want me to host those questions now, or should we wait?
Well, let me ask, so Bill Dunbar has joined us now. Bill, had a problem with your time. What's your situation? In other words, do you want to?
Yeah, equally constrained. I'm afraid I have to go. And my internet wasn't allowing me to start when I when I needed to. We're all slaves to the quality of our internet now. And so I apologize. Maybe you can put me on the next time around. Yeah, really nice to see a lot of familiar faces and friends. And yeah, I do have to go. I have a hard stop. We're pitching another investor for Series B for Ontera, which used to be Two Poor Guys, which spun out, and that's my conflict at 11, which is a nice problem to have. But yeah.
Okay, all right. Well, too bad it didn't work out, but we'll see if we can get you on a future agenda. Thanks.
Yeah, and I'm real sorry. It's just one of those things, you know.
Okay, all right.
But
all right,
thanks.
so let's go back to you and questions for Lou. People, so the way we don't have so many people here, so I think if you just have a question, just unmute yourself. Maybe raise your hand if we get multiples, but who has a question for Lou? Well, I'll ask the first one then. Lou, I'm not clear about to what extent is your Startup Sandbox a UCSC project versus an independent project? It sounds like some of the people coming in are UCSC faculty, some are not. So what is the exact relationship to the campus?
Thank you. So we created what's called an affiliation agreement with the university. And what that basically means is that we're directly affiliated with the university, and we are the commercialization site for the University of California at Santa Cruz. And we run independently from the university. So we've built the lab. We're here for all of you and for whatever IP comes to the Sandbox from the university or from within the UC system as a whole. And the number of companies coming out of UC certainly wouldn't support, the ongoing activities of the Sandbox. So we open it up to additional companies as well.
Okay, but the building itself then is not a UC building per se.
No.
You're really an independent entity.
Correct.
Yeah, okay.
So we have to fund everything on our own. We had the initial funding, and we proved the model. Scott Brandt has been just an exceptional sponsor for us, and was able to secure an additional $200,000, part of it out of his budget, part of it out of the provost's budget, that enabled us to continue through last year. And now we're raising additional funds. So.
Okay. Other questions for Lou? Todd?
Is there revenues coming back to the Sandbox?
So the Sandbox is basically a real estate play, meaning we secure the space, we pay a lease on the space, and then we rent benches, fume hood, BSL2 lab space, and that rental at our 3/4 capacity will give us a break even. There's only one individual that is paid, and that is the lab manager. And we previously had 2 assistant facility and lab managers, and now they've been replaced by 1 lab manager. So everyone else is pro bono.
How many from other campuses of UC are there?
We've had, don't hold me to this, I'm doing it from memory. We've had 10 companies from other UC campuses. We have a very good working relationship with UC Davis and UC Santa Barbara, been very helpful, and UC San Diego.
How many campuses have a similar Sandbox?
That's a very good question. We're working very closely with this Victoria Slivkoff at UCOP, and I think there are only 4 campuses that have incubators such as we have. And in fact, she has hosted Zoom videos where we've given an overview of how we've been able to create so rapidly and so successfully the incubator. And now we're mentoring a couple of additional campuses on growing those incubators, Irvine being one, and in fact, UC Davis being another.
Any other questions for Lou here? Okay.
I'd have to add that in a live presentation that Victoria Slivkoff and her cohorts presented to the UCOP Board of Regents last year. They indicated the University of California system has the largest portfolio of intellectual property of any university in the world. And the regents were stunned. They were absolutely stunned. And that's true. And in fact, you know this better than I do, but the UC Santa Cruz professors that publish are the most cited publications of any university in the world. When I tell people that UC Santa Cruz is in the top universities nationwide in total research grants year in and year out, they're just stunned. $150 million average per year. So there's so much to be proud of the University of California at Santa Cruz, and we are. I want to add one other thing, and I'm talking on, but I certainly have a passion for both UC Santa Cruz and for the capability going forward. I believe that there truly is an opportunity to evolve Santa Cruz, from a tourist destination in an absentee landlord residential area to a bioscience center that's a commercial center and recognized worldwide. And right here in the Delaware corridor, I believe that's possible. And as you know, Chancellor Reeve has renamed what was 2300 Delaware to the West Side Research Park. And we do have plans, and we are engaged with investors to build out 100,000 square foot commercial, it would be Santa Cruz Bioscience Commercial and Manufacturing Center, where companies that graduate from the Sandbox can actually grow their companies here in Santa Cruz. And that's something that most of these companies want to do. Of the first 6 companies, 5 of them wanted to stay here in Santa Cruz because they have the capability of interacting with professors such as yourselves. They continue to be able to draw on the expertise of the university and be able to hire tremendous talent from the university. So I'll stop there. I've gone way beyond your question.
Okay, now, especially since we've are not going to get Bill's talk, we're okay with time. Well, thank you very much, Lou. And so we're going to have an example of one of the projects going on there, and this is Professor Scott Lokey. So Scott got his bachelor's training at Trinity University, which is a small school in San Antonio, Texas, and then went on to get a PhD at the University of Texas. And then he did postdocs at 2 very prestigious places, very different. One is at Genentech, which was kind of the founding big biotech company, and then at Harvard. And then we got Scott recruited to UCSC in 2002. He's a professor in the Department of Chemistry and Biochemistry. So Scott, it's all yours.
Thanks, Barry, and thanks for the invitation. It's an honor to speak to this crowd, and yeah, this is great. All the familiar faces. In fact, Todd Wibke and I had some visions of starting ventures together when I first arrived here in 2002, and that was even before we got into this this area. So yeah, Todd, it's great to see you continuing to support ventures from UCSC. So I want to share. Let's see if you can see this.
Looks good.
Okay. And can you see my full screen now?
Yes.
Mm-hmm.
Okay. So I'm going to try to do this company justice. These are 2 of my ex-graduate students, Cameron Pye and Josh Schwackert, who graduated, got their PhDs on the same day. They gave their dissertation seminars back to back. And they basically started at the same time, and it was kind of fitting because they were sort of the dynamic duo in the lab. I had been working in this area of cyclic peptides, and in particular trying to study membrane permeability in cyclic peptides, and I'll get into the science in a minute. But these 2 guys came in when our understanding of what was going on and the mechanistic ideas were just sort of starting to take root, but they really carried it forward in a big way. And they really didn't know what they were going to do after graduating. But I had been promoting the participation of my students in an annual conference on macrocycles, which is this, sort of narrow field that we're in, but definitely a growing field of growing interest. And this was mainly a conference for industry types, kind of have to pay to play to go to this conference. It's not a really academic oriented conference, but it was fun for me to bring students there just to see, what this world is like. And they ended up talking to a guy who was in a company who had started a company in this area, and he was kind of talking down to them in a way that really irritated them. And they mentioned that they, had some ideas about where this field could go, and he sort of laughed them off like, "Ah, you young whippersnappers, you're so naive." And that really got under their skin enough that they said, "You know what, we can do this. If that guy can do this, we can do this. We can start a company." And so they did. They started a company incorporated a year before they graduated, and had been working out of Josh's garage for a year. And then they had a little bit of a foray into an old teaching lab in Sinsheimer. They got, I can't remember how that was organized, but they got a little space in there, worked for a while in there until they moved into the Sandbox. And the Sandbox, it was a great opportunity for them to actually move into real lab space that was nicely decked out with nice equipment and infrastructure. And that's really where they got going. So let's see, I just wanted to get the details. So my participation in this company, it was very tangential, and I just acted mainly as an advisor to them, because I had already founded a company in this area called Circle Pharma, and was still very much involved in Circle. And that was mainly through QB3 and the Startup in a Box, which is kind of an analog of the Sandbox up at UCSF and through QB3. And so I had some experience in this, but Kim's understanding as CEO rapidly, went beyond what I understood just as a scientific co-founder. But I had to sort of stay at arm's length from UNP because of my involvement in Circle, and they really ended up competing and are still in a healthy competition now. But they raised a round of quote unquote pre-seed funding at the end of 2018, after being in the Sandbox for a year, for $1 million. And then last October, they closed a deal for $6.25 million. They have 14 people in the company now. Half of them are alum from UCSC, ranging from research associates all the way to postdocs. And it's a great company, dynamic company. And I can't say enough about these guys. I love them to death. They're great. It was painful to have to stay at arm's length and to not be able to participate more, because I really believe in their approach, and I think they're going to do amazing things. So just real quick, I think I can spend a few minutes. I don't want to get into the nitty gritty of the science, but just to give an overview of what's exciting about it. The idea is that there are a lot of targets in drug discovery, and that is the proteins that are disease-based, that are responsible for disease. Generally, most often they're proteins. And that's sort of the low-hanging fruit, or it's above the water here, the iceberg analogy, where most of these targets are druggable in that it's relatively easy to find a small molecule that you can take as a pill to inhibit these targets. And it's relatively easy. Of course it's a huge job and very difficult, and takes many years and lots of money. But there's this other class of targets that's under the surface of the water, that is this untapped reservoir of tens of thousands of potential protein targets that are simply unavailable to traditional small molecules, simply because they're inside the cell and they're too large, or they operate by mechanisms that don't involve deep pockets or clefts that small molecules can bind into. And so there's this understanding that if we only could have some kind of other molecular entity that could cross the cell membrane and then block or inhibit the function of these larger protein interfaces, then that would open up this huge untapped reservoir of new targets that are relevant to basically every disease. And so this is really a technology company. It's not just cancer, but they are working on cancer as sort of their preliminary proof of concept. So the idea is that most drug candidates are either too small or too big. That is, very small molecules are great because they can cross cell membranes, but they're too small to hit these undruggable, so-called undruggable targets, the challenging targets. Undruggable targets are classically undruggable targets are relatively easy to hit with or to inhibit with large molecules like antibodies, biologics. But since most of these undruggable targets are inside the cell and antibodies can't cross the cell membrane, these undruggable targets are out of reach, the ones inside the cell, to the antibodies that could otherwise inhibit them if they were outside the cell, which antibodies are very good at doing for those targets that are reachable, that are not inside of the cell. So there's this middle ground. There are molecules that can do this, and these are macrocycles. They're cyclic in nature. That is, they're kind of represented by a string of atoms that are then cyclized like a necklace so that they form a cyclic structure. And because of the nature of how they're constrained and their relative complexity compared to small molecules, these macrocycles can do things that small molecules can't do. They can bind to these undruggable proteins. And then the trick is getting these macrocycles into the cell. And so there are a large number of companies that are extremely successful who have designed macrocycles that can hit just about any target. And they're very good at finding these macrocycles that are able to hit most of these undruggable targets. The problem is they can't seem to be able to get them inside the cell. And so that's where all of these companies have come up short. But UNP, and I'm not going to go into the details here, this is their pitch deck, and it's cool. I'm probably not doing it justice, but this is really a continuation of my research. That it's basically, they're taking the vision that I had early on in this area and making it a reality. And they're very talented, and if anybody can do it, they can. And I've actually, it's so pleasing. I guess it's kind of like when you're a parent and you see your kid go and do something that you never could have done, and you have this kind of mixture of, envy, but also mostly pride because they're succeeding in carrying something beyond where you even could envision. So last time I talked to them, they were explaining to me these in silico design processes that they're going through to take their hit compounds and convert them into permeable, that is, compounds that can cross the cell membrane, and going through these cycles, iterations of design and synthesis. And they're doing it with so much finesse and with really cool technology that I never could have imagined was possible. Hang on, Xander. So this is the data that they show, how they go through these design cycles and iterations. And I'm giving a talk right now. Okay. And if you go, I'll talk to you. Okay. And yeah, school has not started yet. It will start.
Nope.
And so, say hi. Yeah, so that's Xander. He's in first grade. He's doing first grade by Zoom. It doesn't work all that great. You're in a break. Here, okay, you know what? I'm about to take a break, Xander. Here, let's go into the kitchen. You know what? I am actually done right now, other than to say that I have a lot of a huge amount of confidence in UNP. I think they're going to be incredibly successful, not only because they have a great technology and a great idea, but also Cam and Josh are just a super team and are very, very smart and are great leaders. It couldn't be a more perfect fit for them to have gone into this venture, and it really wouldn't have happened without the Sandbox, or it would have been much more slow to evolve. They were sort of flailing around in the garage and in the basement of Sinsheimer, and you know, with the Sandbox, they really did graduate to a real lab, and that was provided the necessary infrastructure and support for them to really get that first seed round, and then, you know, the sky's the limit from there. So that's my little spiel, and I'm going to escort Xander out of the room here, and then I'll be back to take any questions. Okay.
Okay.
I'll be
Okay.
right back.
Thanks, Scott.
In,
Right. So I'm going to, okay, I can bring it back so we can see everybody. Ah, there's life in our world today, folks. All right. So Bruce Margon had a question of Lou. So those of you who don't know, Bruce was Vice VP for Research at UCSC for a number of years. Bruce, your question?
Thanks, Barry. Can I be heard and seen?
Yes.
Okay. So it's a question for Lou, or possibly for any of the speakers today, which is, most research universities have a standard menu of reasons for commercializing intellectual property, including UCSC, of course. And these are things like we enrich the intellectual environment of the campus, we enhance the university's reputation, we can contribute to the local economy, we give students real life experience, make them more employable. But on that menu, inevitably, is also that there can be positive financial remuneration for the campus's investment in IP, and in particular, that the campus can recognize real cash benefits from licensing fees and royalties. So I noticed that Lou, for example, said we're bringing products to market very quickly, and so I'm wondering, is there any finite example now of the university's investment of money in IP that has returned some positive financial benefit to the campus?
Well, certainly David Deemer is on the call, and the licensing of the initial nanomore technology to Oxford Nanopore is one example. David, is there anything you'd like to say about that?
He might not be with us anymore, I guess.
Dave,
Okay.
yeah, Dave down there. Dave, you can unmute yourself.
You just
Okay, I
did.
did. Can you hear me?
Yes.
Yes.
Okay, good. Yeah, nanopore sequencing was basically developed at UC Santa Cruz starting in 1997 when Mark Akeson joined my lab. I was still in the chemistry building at the time. And over about a 10-year period, we got up to the point where the patents that we had developed were attracting the interest of companies. One of them, Oxford, what is now Oxford Nanopores, came to the campus in 2007, visited me in my office, Mark was there as well, and basically said, we want to license all of your nanopore patents. We think this is going to go someplace. So it's a big risk for a company. And we learned a lot of lessons interacting with this really wonderful company. And I got a little talk I'm going to give, I think next month to the Emeriti again, when we have those little 10-minute things. I'm going to go through the lessons we learned. But one lesson that was very important was that an academic like myself and Mark can get a deal with a company like Oxford, which was a startup company back in that time period, 2007. And suddenly things begin to happen that we can only dream of. You know, academics are happy if they get $1 million for a grant. This company had 100 million pounds to start with. We have a few grad students, maybe a postdoc in our lab. They had 100 dedicated employees, and they did stuff for nanopore sequencing that I don't think any other company could have done. They, instead of a single nanopore, which Mark and I had been working with for years, they came up with the MinION, which had 2,000 individually addressable nanopores. They had a membrane that was able to be shipped through the mail and last for months of time in somebody's lab. And right now, they're doing something called the LAMPORE, which is specifically addressed to using nanopore sequencing for the COVID virus. It's being tested in England. The English government has given 50 million pounds of support to develop the nanopore. We hope to get the first LAMPOREs in this country on this campus in the very near future, as soon as they're ready to do that. The IP, by the way, is valuable for the campus. The inventors get about a 1/3 share of royalties and fees that come in. The rest of it, a pretty good chunk of it, goes to the campus. So when I've talked to Jeff and some of the people in the tech transfer office, they are very happy with what is coming in from Oxford Nanopore, as they have grown from a small company, $100 million, to a major company now valued at $1.5 billion. So they're still private. They're considering an IPO in the near future. And it's just been a wonderful ride. If you want to hear more about it, tune in sometime next month and I will give you my 10-minute spiel of all the lessons we learned as we went along with Oxford Nanopore.
David, thank you so much.
I'm sorry, I didn't realize you were on the call. And of course, I'm very familiar with that situation, that I co-signed your initial IP agreement with Oxford Nanopore.
Yeah.
You're right. It's a wonderful example of a huge intellectual achievement. But I hate to sound venal, but I will say that the royalties accrued from them thus far have not come even close to the total amount of money that UCSC has spent on IP commercialization. On the other hand, I don't regret the arrangement for one instant, because it has been an incredible intellectual success, just as you have said.
Let's just look at a couple of others. You know, cruise foam, based on the work that Marco Rolandi did at the university, you know, they're just getting started in the area of foam packaging to replace the petrochemical foams that are rampant in our environments today. But you know, I think we need to look forward at technologies such as cruise foams technology and what that potentially will return to the university. And I would also say that the university is just beginning, it's in its infancy, on commercializing intellectual property out of the university and finding a significant reward for that effort. So I think we look forward and I think go forward with the confidence that this will, over time, create a significant endowment for the university.
Okay, thank you. So questions for Scott. Scott's back with us here. Question on Scott and his company. Todd?
Do you envision this company as a consulting company, or will it actually develop its own drugs?
They're doing both. I guess, I don't know, the relationship, they have a pharma partner right now, a big pharma partner that they're working toward on a collaborative research project, and you know, UNP would own the IP, but that would be, you know, licensed by that company if anything hit. Then they're also doing their own internal discovery, and I think they're mainly focusing on that. So, and I imagine they would license out any promising candidates if they get, you know, good preclinical data on them. So I think it's a hybrid model.
Okay, I had a question on the science, Todd, and that is this company focused on the drugs making the new macrocyclic compounds? Or you mentioned that the key problem was getting them across the membrane.
Yeah.
Are they addressing that membrane crossing phenomenon?
Yeah, it's a combination of an interesting technology that allows them to synthesize hundreds or even thousands of these compounds in one round. That allows them to rapidly test a lot of hypotheses about what it takes to cross the membrane and maintain binding or biological function. And since those two are sort of separate issues, it's really hard to find compounds that are both permeable and have a biological function. So you have to make a lot of compounds in order to sort of understand those relationships, and then they're using a computational approach to derive the relationships where you can't really analyze 1,000 data points sort of manually, but it's really a combination of their synthesis and their design approach that I think is their sort of secret sauce.
Okay, other question, Karen.
Yeah, thanks, Barry. Thanks, everybody. It's been very enlightening. This is completely out of my area, of course. So the question is more at a larger level, and that is, do you see any downsides to commercialization as incentivizing research on the campus? Any downsides or any, sort of generic issues that speak against it, or yeah. I'm going to mute myself now.
I guess.
Yeah, I don't know if that's really a question for me. Maybe that's one for Lou or others.
I'm happy to address the question.
Mm-hmm.
First of all, I think we need to acknowledge the tremendous advances that the professors at UC Santa Cruz have made over the years in basic and applied research. That's just fundamental. And it goes all the way back to Provost Sinsheimer in 1978 and the transition from a preeminent liberal arts school to a preeminent research university. So one, I think I personally and we at the Sandbox first and foremost respect that level of excellence, and that based on that level of excellence, the question is, can and should these areas of basic and applied research that have the potential to change people's lives in a very positive way, and to change the health of our planet, quite frankly, should those be commercialized? And I think that's a relevant and reasonable discussion to engage at amongst the professors at the university. Does it harm the overall basic and applied research capabilities, or does it actually enhance them? And I think those are the conversations, not for me to lead, but I think for the university to engage in, because there's such incredible excellence in that area, and you're the custodians. We're here to facilitate whatever decisions are made.
Okay, Dave, did you have a question?
Yeah, I did. And also a comment. I'll do the comment first and then ask a question of Scott. The comment is that I've been involved with the Startup Sandbox for about 2 years now. And something that hasn't been brought out is that Lou has welcomed educational activity. It's much more than just business stuff, I've had 2 interns, one a summer intern from the biomolecular engineering department. I've had another intern from Australia working in the laboratory. I've had 2 high school students getting started on their high school science fair project. Lou has welcomed that kind of educational activity. I think the more of that, the better. So that's my comment. A question to Scott has very simple, and this might be proprietary, but I'll ask it anyway. Do you have a target protein, intracellular target protein, that one of these microcyclics could potentially inhibit, for example, if they get into the cell?
Yeah, that would be, so I think that the targets that UNP is working on are proprietary, but, or at least I'm not authorized to disclose them. But in general, I think if you look at sort of the protein-protein interactions that are involved in cancer progression, tumor suppressor proteins, and other signaling molecules that don't have, that are, helices binding into grooves like, BCL2, BCLXL, those kinds of proteins. So yeah, it's sort of the usual suspects as the preliminary, as sort of the proof of concept. And then you sort of end up looking at more obscure targets. But there's a lot of interest in these kind of generic protein-protein interactions in cancer as kind of the proof of concept.
I just have a quick follow-up on what you're saying. Our little company was called UpRNA, and we were trying to make siRNA, which can inhibit protein synthesis if it can get into the cell.
Yeah.
Last year, an extraordinary result came out, published by a group back east with help from Roche Pharmaceuticals, as I remember. They managed to get siRNA targeted for the protein called huntingtin, which is the toxic protein that accumulates in the cells of Huntington's disease.
Yeah.
Huntington's disease is invariably fatal.
Yeah.
You may remember that Woody Guthrie died of this. You might not know that his mother died of Huntington's and two sisters. It's a genetic disease. It's just a terrible disease. You're gone in your 50s if you happen to have that particular gene. As a result of injecting this small siRNA into the spinal fluid of Huntington's patients, they could turn down the synthesis of huntingtin, this protein that accumulates otherwise. And that's just an example of how powerful an intracellular delivery could be. And more power to Josh and Cam. I was with them in the lab for about a year. And it was really fun to watch them go to work, and they were very loud. We had to kind of put up with a lot of loud conversation as they were having fun with their project. Olaf wanted to say something. Olaf wanted to say something.
Hi, Olaf.
Hi. It's not a question for you. It's actually picking up on the question that Karen had.
Yeah.
About the, are there any downsides? I don't think there is so much, this is just my personal opinion, that there is so much downside for having incubator space for, faculty. But there is, I've always thought that there is some risk to renting out space on the campus because of the monetary sort of incentive of renting that out to outside groups, perhaps at the risk of not providing space to faculty. So I just would like to put that out there. I think incubator space off campus is different, but there is the sort of incentive if people come in from the outside that they pay for the space, whereas there might be faculty in various departments that would like to have extra space, but maybe would not get that space because of other people occupying it. Anyway.
Okay.
Yeah, and if I could just add, I think my experience has been with my postdoc at Genentech and working, co-founding this other company, Circle, and then working with a lot of companies through the years, is that the scientists involved in the companies generally remain focused on the exciting science that they're doing, and making money is usually secondary. I mean, it's nice to make a lot of money, I think, but and I'm sure Cam and Josh would like to have that compensation for working 80-hour weeks for the last 4 years, but really their passion is still in the science, and it's kind of an academic crew, even in industry. They're sort of oriented in that direction. The thrill of discovery is kind of still kind of foremost.
Scott, I'd like to ask you, what impact have you noticed on your own research at the university as a consequence of your involvement with different companies or startups?
Yeah, I think it's given me a broader and deeper perspective on drug discovery, because the real world of drug discovery is very complex, and there are a lot of landmines in terms of trying to optimize not just the sort of biochemical potency of a compound, which is really where most academics are focused when it comes to lead compound discovery, but also the properties and whether it'll be orally bioavailable and how fast it's metabolized and all of those things. Academics in the chemical biology arena, medicinal chemistry arena, don't often think about. So it's given me a much broader perspective on drug discovery, and also it's affected my ability to help my grad students and postdocs find jobs in industry if they want them. I've gotten a lot of contacts in industry, and so I've allowed them, or even just their participation in meetings with the companies that we collaborate with has just kind of given them an edge and given them sort of an inside perspective into industry that they wouldn't have had otherwise.
I do have a follow-on comment, if there's time for that, Barry.
Yes.
So David mentioned mentoring two high school students, and that in and of itself has created an opportunity that now is being formed, and that opportunity is that there truly is a capability that will start locally, and it's being sponsored by one of our Founder Circle members, excuse me, to create an opportunity for middle school and high school girls to engage in research projects at the Sandbox under the mentorship of either postdocs or professors who can really work with them on forming a viable research project. And the vision of this is to democratize research, and beginning with girls in middle and high school. What a wonderful thing that can take place here. David, you didn't mention, but Michelle Nazareth, who you mentored on her research a year ago, went all the way up to the nationals and to the Intel bake-offs, and was actually interviewed by National Geographic last year. And this year she's doing something that is quite profound, that David would be able to describe to you better than I can. And now this new young girl is a sophomore at Monte Vista High School, and David is mentoring on another profound research capability that could lead her to a career, of course, in research, and pre-qualify some of these individuals to be students at the University of California, Santa Cruz someday. So it's a wonderful opportunity, and research in and of itself is what we sponsor at the Sandbox. Commercialization of that research is a byproduct of the basic and applied research that comes to us. So I don't know if there's anything else that anyone wants to say around that, but I wanted to point that out.
Well, thank you, Lou. So any other further questions or comments? Corinne?
Do high school counselors know about this so they can steer high school seniors to apply at UCSC? And is there anything you can do to make sure they know about the opportunities here?
I think that's directed at me. Is that correct?
Mm-hmm.
Okay. So what we will do is start locally, and we'll be engaging the local high schools to let them know of the opportunities that they would have for their girl students. And we specifically are starting with girls. That was the intent of the individual that is sponsoring this. And obviously it will broaden out over time, but he definitely wants to start with girls. So we will go to the counseling groups and to the the science faculties at the local high schools, and we'll begin there.
Okay. All right. Well, I think we will wrap it up. A little bit shorter than planned, but because of Bill Dunbar's conflict, we weren't able to have his talk. So our next gathering will be the third Thursday in October, and that will be the 5 short presentations of current emeriti, what they're doing in 5 very different areas. And then we will meet again in the third Thursday of November. And don't forget Joel's talk, which will also be in November. So good to see everybody. And thank you for your participation. Thank you, especially Lou, and thank you, Scott.
Thank you. It was fun. Thanks a lot.
Thank you. What a privilege to be invited. Thank you very much.
Nice to meet everybody.
All
Take
right.
care.
Okay. Thanks to all. Okay.